Mendel's experiment on dihybrid cross. A cross which involves two pairs of contrasting characters is called a dihybrid cross. Mendel continued with his experiments by involving more than one pair of contrasting characters at one time. This time, he crossed pea plants having round and yellow seeds with plants bearing wrinkled and green seeds. In this experiment, since there are two pairs of contrasting characters, namely seed shape and seed color, therefore it is called a dihybrid cross. Let us understand about the dihybrid cross of pea plants involving round and yellow seeds with wrinkled and green seeds. Parental generation, P1. In Mendel's dihybrid cross experiment, he chose pea plants having round and yellow seeds and those with wrinkled and green seeds as parent plants. The genotype of the plant with rounded yellow seed is capital R, capital Y, and the plant with wrinkled green seeds is small r, small y. Here, round shape and yellow color of seed are dominant characters, while wrinkled and green color of seed are recessive characters. Now let us study the gametes formed from the parental generation, P1. When the gametes of the P1 generation are formed, each pair of genes segregates independently from each other, which means that capital R, capital Y would not produce capital R, and capital Y gametes. It would produce only capital R, capital Y gametes. Similarly, the plants with small r, small y would produce only small r, small y gametes. Thus, we can conclude that in gametes, a pair of genes are represented by only one gene. Let us study the result of Mendel's dihybrid cross in the next generation, that is, the first filial generation, F1. First filial generation, F1. On the basis of the experiments with monohybrid crosses, Mendel expected that only the dominant characters would appear in the F1 generation. Thus, he anticipated that the plants of the F1 generation would have yellow and round seeds. His prediction was right. Though the genotype of the pea plants of the dihybrid cross F1 generation were capital R, capital Y, small y, but phenotypically, they were plants with round yellow seeds as capital R gene, for the round shape was dominant over the small r gene for the wrinkled shape. And similarly, capital Y gene for yellow color was dominant over small y gene for green color. The plants of the F1 generation of dihybrid cross produce four types of gametes: capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Of these, the capital R, capital Y, and small r, small y gametes are similar to the gametes produced in P1 generation. When the F1 generation plants undergo self-pollination, they give rise to the second filial generation, that is, F2. The four types of male gametes and the four types of female gametes give rise to 16 different possible combinations. Let us understand how characters are transmitted with the help of Punnett square or checkerboard. On the top of the chart, male gametes are arranged, while at the side of the chart, female gametes are arranged. In the boxes of this chart, 16 different possible combinations of the F2 generation have been shown. Let us now understand about the second filial generation, that is F2, in detail. Second filial generation, F2. Second filial generation F2 is formed through the selfing of F1 plants resulting 4 phenotype and 9 genotype combinations. First, let us determine the phenotype ratio. Round yellow phenotype of the seeds is observed in a total of 9 squares on the checkerboard. They are numbered 1, 2, 3, 4, 5, 7, 9, 10, and 13. Wrinkled and yellow phenotype of the seeds is observed in a total of 3 squares on the checkerboard numbered 11, 12, and 15. Round and green phenotype of the seed is observed in a total of 3 squares on the checkerboard numbered 6, 8, and 14. While the wrinkled and green phenotype is observed in only 1 square numbered 16. Thus, the phenotype ratio of the F2P plants is 9:3:3:1. Now let us determine the genotype ratio of Mendel's dihybrid cross. There are 9 different genotypes. Genotype R, capital Y is observed in only 1 square in the checkerboard numbered 1. Genotype R, small y is observed in 2 squares in the checkerboard numbered 2 and 5. Genotype R, small y is observed in 1 square in the checkerboard numbered 6. Genotype R, small r, Y is observed in 2 squares in the checkerboard numbered 3 and 9. Genotype R, small y is observed in 4 squares in the checkerboard numbered 4, 7, 10, and 13. Genotype R, small y is observed in 2 squares in the checkerboard numbered 8 and 14. Genotype r, capital Y is observed in 1 square in the checkerboard numbered 11. Genotype r, capital Y is observed in 2 squares in the checkerboard numbered 12 and 15. While genotype r, y is observed in 1 square in the checkerboard number 16. Thus, the genotype ratio is 1:2:2:4:2:1:2:1. The square numbered 1 having genotype R, Y is pure dominant with round and yellow seeds, and square numbered 16 having genotype r, y is pure recessive with wrinkled and green seeds. We have studied dihybrid cross between rounded yellow seeds and wrinkled green seeds and monohybrid cross between tall and dwarf plants in detail. What will be the results of monohybrid cross between round and wrinkled seeds? Let us find out. Unlike the monohybrid cross between tall and dwarf plants, all plants in F1 generation will have round seeds because rounded seed is a dominant character over wrinkled seed. By selfing of these hybrid plants, we will get 3 plants with rounded seeds and 1 plant with wrinkled seeds. Out of 3 plants with rounded seeds, 2 are hybrids. Hence, phenotype ratio in monohybrid cross in F2 generation is 3:1, while genotype ratio is 1:2:1. After learning about Mendel's principles of heredity, let us learn about the disorders caused due to disturbance in hereditary mechanisms.